Here is an illustration of the salmon life cycle. We begin with fertilized eggs. These develop into what are known as eyed eggs, in which you can see the developing embryo. The eyed eggs will eventually hatch, releasing alvin. The alvin are little fish with very prominent yolk sacs. Eventually, the yolk sac is resorbed. Once the yolk sac is resorbed, the fish are known as fry. The fry grow into parr, and when the parr are ready to move back into the saltwater environment, they undergo what is known as the parr-smolt transformation. When they're smolts, they can survive in the marine environment, and from smolts they develop into adults. Eventually, the adults return into the freshwater environment where they migrate upriver to spawn, thus completing the life cycle. Hatcheries are where the salmon are reared from egg to smolt. There are two types of hatcheries. We have production hatcheries, where the fish are destined for salmon farms, and then we have hatcheries that are designed for the restoration and enhancement of wild salmon. Hatcheries were originally designed for the latter. Production hatcheries tend to be located near the intended farm and use local broodstock to prevent the introduction of foreign animal diseases. The purpose of the wild salmon hatchery is to recover depleted fish stocks, wherein hatchery-reared salmon can make up 25% of the harvest. Wild salmon hatcheries are also intended to improve the survivability of specific salmon runs. This is a remedy for damaged freshwater habitats. Wild salmon hatcheries use locally adapted broodstock to improve survivability. The salmon are reared and released on site to improve returns. Low rearing densities are used to improve survivability, and interbreeding is kept to a minimum to preserve adaptive traits. Incubation and rearing facilities are designed to mimic the natural environment, which facilitates the development of natural behaviors. For wild hatcheries, nets are used to catch wild salmon as they migrate upriver to spawn. The eggs and sperm are collected, and the eggs are fertilized. Hatcheries reduce the risk of predation and maintain optimal conditions for early development. During the riverine phase of the outmigration, predation can account for the death of over 70% of the population. Young salmon are reared in hatcheries until they are ready to smolt, at which point they're released at the mouth of the river. In so doing, you can skip the riverine phase and reduce the risk of predation. Some of the hatcheries are placed further upriver to keep the population isolated in order to preserve adaptive traits. These populations experience lower rates of survivability due to higher rates of in-river predation. Development begins in the incubation box. The fertilized eggs are kept in these boxes until they develop into eyed eggs and alvin. The developing eggs are given a constant supply of fresh water to maintain optimal levels of oxygen and temperature. Non-viable eggs are removed in order to prevent saproligniasis. This is a condition caused by the oomycete saprolignia. If saprolignia were allowed to grow on the non-viable eggs, it could quickly spread to the viable eggs, reducing survivability. Once the yolk sac is resorbed, the alvin become fry, which are transferred to rearing troughs. At this point, the fish are transitioned to pelleted feed. They remain in the rearing troughs until they become parr. Parr are transferred into rearing tanks, where they can develop into smolts. Rearing tanks can be raceways or recirculating systems. Pre-smolts are vaccinated before they're transferred into marine pens. Wild hatchery-reared salmon are not vaccinated. The rationale for this is unclear, but perhaps it's to avoid giving the hatchery-reared salmon a competitive advantage over the salmon that spawned naturally. Things to consider before vaccination include the species of fish you plan on rearing, given that certain vaccines are only effective in a narrow range of species. Other things to consider include the duration of protection offered by the vaccine, the route of administration, and the cost effectiveness. If the cost of the vaccine is higher than the value of the fish, then vaccination is pointless. Prior to vaccination, it's important to minimize stress and to deprive the fish of food. Only healthy fish should be vaccinated, and you need to make sure that the vaccine is timed appropriately so that the fish have sufficient time to develop immunity before they're exposed to pathogens. There are several types of vaccines in aquaculture, but some of the more common ones include bacterins and modified live vaccines. Bacterins are pathogenic bacteria that are killed in formalin. Examples of bacterin vaccines include Aeromonas salmonicida, Yersinia ruckeri, Moritella viscosa, Ranibacterium salmoninarum, and Picerichetsia salmonis. Modified live vaccines include vaccines against bacteria and viruses. These organisms are grown in culture so that they become attenuated and less pathogenic, while still eliciting a pathogenic response. Examples of modified live vaccines include ISAV, Flavobacterium psychrophyllum, and Edwardsiella ictaluri. Vaccines can be administered in a variety of ways, including oral administration, immersion, and intramuscular or intracelomic injection. The method used is dependent on a variety of factors. Oral vaccines are quick and easy to administer. Moreover, they're relatively stress-free. However, they provide little and often short-lived immunity. Another challenge associated with oral vaccination is in preventing inactivation of the vaccine within the gastrointestinal tract. Examples of oral vaccines include Picerichetsia salmonis and ISAV. Immersion vaccines are easy to administer and cause minimal stress. They provide effective but short-lived immunity. Smaller fish are given dip vaccines, which is a highly concentrated vaccine solution. Larger fish are given bath vaccines. Such vaccines are lower in concentration, so they require a longer duration of exposure to be effective. Examples of immersion vaccines include Yersinia ruckeri. Injectable vaccines can be administered in the muscle or the coelom. Injectable vaccines are labor-intensive and very stressful for the fish. The injections can be given manually, and highly trained personnel can administer well over 1,000 vaccines per hour. As technology advances, new methods of administration become available. With large-scale production, there are fully automated systems of vaccine administration. Injectable vaccines provide strong immunity, and multiple antigens can be administered per vaccine. However, there are certain challenges associated with injectable vaccination. Firstly, the fish need to be anesthetized. There are also size limitations, so if the fish are too small, vaccination must be delayed. Injectable vaccines can also cause localized tissue reactions. They can even be associated with secondary bacterial infection. Pre-release health checks are conducted on smolts before they're transferred to sea. The transfer will only occur when the fish are shown to be disease-free. Health checks include histopathology, PCR, and bacterial culture. It's worth noting that pre-release health checks are not performed on wild salmon reared in hatcheries. The smolts are transferred via wellboats, which are large ships that are specifically designed to host live fish. Think of them as mobile aquariums. The transfer of fish from the freshwater to the saltwater environment is very stressful. This can result in failed smolt syndrome. The syndrome is characterized by a failure to feed despite having an appetite. The affected fish exhibit low levels of activity, and they cannot compete with normal individuals. As a result, they have stunted growth, which can progress to emaciation. The cause is unclear, but there have been a number of associations through controlled studies. Fish that are kept longer pre-transfer seem to be more resistant. There are also fewer cases when the transition is gradual. For example, if the fish are moved from a freshwater tank to a saltwater tank before they're moved into the ocean, there are fewer cases of failed smolt syndrome. If the fish experiencing this syndrome are moved back into the freshwater environment, they immediately resume feeding. This syndrome has also been associated with underlying diseases and other causes of stress, like saproligniasis. In order to prevent failed smolt syndrome, you need to minimize stress and ensure the health of the fish. You should also reduce the variability in fish size to prevent intraspecific competition. How you feed your fish is dependent on the type of system you're operating. In pond aquaculture, fertilizers are commonly used. Fertilizers promote the growth of algae and plants. These primary producers bolster the food pyramid, resulting in a stronger population of zooplankton, snails, arthropods, tadpoles, frogs, and smaller fish. Types of fertilizers include manure, urea, and ammonium phosphate. Natural feeds can also be used in pond aquaculture. Common protein sources include fish meal, trash fish, and snail meat. Common sources of carbohydrates include rice or corn bran, coconut meal, and rice mill sweepings. As a source of lipid, oil cakes are used. These are the residues that are left over from the production of various oils, like palm oil and sunflower oil. Commercial feeds are also used, which are pelleted formulated feeds specific for the fish you're rearing. Proper feeding regimes are necessary. Any unconsumed feed will accumulate along the bottom of the pond, reducing water quality. This will also result in decreased levels of dissolved oxygen via bacterial growth and decomposition. Aquarium fish have a variety of food types, the most common of which are flakes and pelleted feed. Flakes are generally of lower quality, and they have a lower shelf life. They're great for surface feeders, but they break down quickly. Furthermore, fish feeding along the surface have a tendency to swallow air, which can cause buoyancy issues. Pelleted feed is generally higher in quality, and it has a longer shelf life. Pelleted feed is also formulated for specific species. Pellets can be variably sized depending on the species of fish and the age class. The pellets will also vary in their density to either sink or swim. It's good for surface feeders, pelagic feeders, and benthic feeders. It also breaks down more slowly, giving the fish plenty of time to eat it. Ingredients for flakes and pelleted feed include algae and plant meal derived from wheat and soybean, animal meal from fish, arthropods, and mollusks, various oils from fish and soybean, and vitamin supplements. Other types of feed for aquarium fish include frozen cubes and live food. Frozen foods are generally made from various kinds of invertebrates which are preserved whole. They're highly nutritious but more expensive. One of the advantages of using whole animals is gut loading. Gut loading is what happens when the invertebrate feed is fed before it's harvested. So not only are the fish getting nutrients from the invertebrate, they're also getting nutrients from within the invertebrate's gut. Frozen foods can also be stored for an extended period of time. Freezing also prevents nutrient breakdown. Frozen foods also tend to be high in carotenoids, which brighten the fish's colors. Examples of frozen foods include bloodworms, brine shrimp, and daphnia, which is pictured on the bottom right. Live food is also highly nutritious, but it's much more expensive. Examples of live food include tubifex worms, crustaceans, and feeder fish. Surprisingly, feeder fish make really good pond fish, and they're much cheaper than koi. Now, for the most part, you could get by with pelleted feed, but there are a number of situations where you might want to consider using live food. Live food is really good for breeder fish, given that they have higher nutritional demands. It's also good for predators and picky eaters, like lionfish. Live food can also provide environmental enrichment. Salmon nutrition has received a lot of attention and has been researched extensively. The feeds are generally pelleted and formulated for different age classes. Pelleted feed must be stored in dry areas to prevent food spoilage, which can occur via bacterial or fungal growth and oxidation. Given that salmon are destined for human consumption, salmon feeds are also screened for heavy metals and other contaminants. Modern salmon feeds are 70% plant-based, made from wheat, corn, algae, soy, sunflower, rapeseed, and broad beans, among other things. This plant material is an excellent source of proteins, carbohydrates, and oils. 30% of the feed is animal-based. This can include raw materials like fish meal, fish oil, chicken meal, and pork meal. Additional supplements include omega-3 fatty acids, eicosapentaenoic acid, and docosahexaenoic acid. These fatty acids are very important for fish and human health. Fish meal and fish oil is derived from fish heads and other byproducts that were not intended for human consumption. Other sources include reduction fisheries, like anchovy. Given that fish meal and fish oil are in limited supply, plant and animal-based substitutions are used. This can include genetically modified canola to produce EPA and DHA. Other supplements include vitamins, minerals, amino acids, and pigments like carotenoids. One of the most important supplements is astaxanthin. This is a naturally occurring carotenoid found in algae, shrimp, lobster, crab, and salmon. This is what gives the salmon its characteristic red color, which is aesthetically pleasing. It's also an antioxidant and a source of vitamin A. It strengthens the immune system and protects the tissues, resulting in a higher quality product. Diagnostics is one of your greatest tools in aquaculture management. Information can be gathered in a variety of ways. Environmental analysis is an obvious place to start, where you can check your water parameters like pH, salinity, temperature, and dissolved gases. You can even check for the presence of certain toxins. Clinical presentation is another important source of information. Things to look for include a change in behavior, a change in appetite, or a change in respiration. Although many of the clinical signs are nonspecific, by monitoring for changes in behavior, you may be able to identify changes in the water parameters. For example, you may notice that a lot of the fish are crowding around the water intake. This is often indicative of low dissolved oxygen. Biometrics are used to monitor growth and development. One of the most common measurements is the fork length. Now, the fork length is the distance from the tip of the snout to the fork of the tail. The image on the bottom right illustrates some of the other measurements that are used and why it's important to know your external anatomy. The gross exam can include an external exam, a skin scrape, a gill biopsy, and a necropsy. Many of the changes noted on an external exam are fairly nonspecific. Skin scrapes and gill biopsies can help you identify specific etiologies. They are a low-cost alternative to histopathology, and they're fairly simple to perform. Necropsies will provide you with a lot of information and the opportunity to take additional samples for ancillary testing. Although gross observations can provide you with a lot of information, it's always a good idea to make use of your histopathologist. They could find an underlying disease that you weren't even aware of. Other ancillary tests include immunohistochemistry, in situ hybridization, and special stains. You can also perform PCR assays for specific etiologies or request bacterial, viral, or fungal culture. Skin scrapes and gill biopsies are an excellent diagnostic tool. They're low in cost and have a rapid turnaround time. However, the fish has to be sedated. For skin scrapes, the skin is scraped by using the edge of a glass slide. By scraping the skin, you can extract a sample of the cuticle and mucus layer. Add a drop of water, even out the sample, apply a cover slip, and look under the microscope. You should be able to identify most parasites based on their morphology and pattern of movement. Gill biopsies are a little bit more invasive. Using suture scissors, clip the tip of a gill filament. Suture scissors are preferred because the cutting edge forms a little pocket that holds the sample in place. Place the sample on a glass slide, add a drop of water, and apply a cover slip. The cut filament should stop bleeding fairly quickly. Treatment requires a multifaceted approach. Chemical intervention should be considered a last resort. It's better to address predisposing factors and underlying causes. Most diseases in aquaculture are associated with husbandry-related issues. Issues can include temperature, water quality, population density, and feeding regimes. The route of drug administration will vary depending on the situation. Certain enclosures are ill-suited for environmental treatment. For example, with cage and pen aquaculture, administering medication into the environment may be ineffective because you're dealing with a large volume of water and the currents will just take the medication away. Water chemistry is another factor worth considering. Certain drugs, like oxytetracycline, are chelated by calcium. so if oxytetracycline is administered into a saltwater environment, it's rendered useless. For this reason, oxytetracycline is added to the feed. The oils in the feed partition the drug and prevent chelation. When planning treatments, you should also consider host factors like fish size, species, and health status. Certain disease conditions are associated with inhabitants. If the medication is administered in feed, the fish that need it most are unlikely to get the appropriate dose. The quarantine tank and the hospital tank are meant to prevent the spread of disease. These tanks operate on a separate recirculation system. When affected individuals are separated, they're easier to monitor. Smaller tanks also reduce the amount of required drug. By isolating the affected individuals, you can also avoid medicating the unaffected individuals. This can be an issue because certain medications can kill cohabitants, like invertebrates and nitrifying bacteria. Quarantine tanks are also devoid of substrate. This prevents the chelation of medication, and it also ensures that pathogens are unable to persist in the environment. One thing to remember is that small tanks are more difficult to manage in terms of maintaining appropriate water parameters, like temperature, dissolved oxygen, and ammonia. Topical medications are generally used to treat external infections, like fungal infections. Sedation may be required. One of the benefits of topical medication is that you could administer high concentrations whilst minimizing drug waste. The treatment is also fairly localized and specific, so you can avoid adverse systemic effects. An example of where this may be necessary is in treating ocular infections. Environmental administration is another means of treating external infections. Typically, this method is used when treating large numbers of fish in a small volume of water. These fish are treated over an extended period of time. The dose depends on the system volume and the target concentration. Bath treatments are very similar to environmental treatments, except they're more useful when treating fish in a large volume of water. First, the fish is removed from its normal enclosure and placed in a smaller volume of water. The treatment is often of short duration, using a higher concentration of medication. As with environmental treatments, baths are used to treat external infections. Enteral administration is useful when treating internal infections. Oral medications are easy to administer and stress-free. However, they are ineffective if the fish is inhabitant. Parenteral administration is also useful in treating internal infections. However, sedation is required. For this reason, it's less feasible when treating large numbers of fish. The medication is generally administered in the muscle or the coelom. This can be challenging because the needle must first pass between the scales. Another disadvantage of parenteral administration is that the medication can have adverse systemic effects. Some medications can also cause localized tissue necrosis. As a result, you need to be sparing in your use of injections and avoid injecting the same spot twice. When treating fish for parasitic infections, there are certain parasites that can be dealt with by interrupting the life cycle. This can be achieved in a number of ways. When fish are well fed, they tend to avoid live prey, which can be host to the infective stages. Other methods include removing the intermediate host from the environment, treating the substrate for free-living stages, and using a UV filter to kill free-living and infective stages suspended in the water column. Once a fish is infected, the most basic means of treating an external infection is through the use of a saltwater or a freshwater bath. This can be used in food fish and ornamental fish. Baths are commonly used to treat protozoal infections and oomycete infections. Freshwater baths are used to remove saltwater parasites like Cryptocaryon, and saltwater baths are used to treat freshwater parasites like Ichthyophthirius multifilus. Baths can also reduce osmoregulatory stress, so it acts as a mild sedative. If a saltwater fish is put into a freshwater bath, less effort is required for osmoregulation. The same is true for a freshwater fish that's put in a saltwater bath. Saltwater also promotes mucus production in freshwater fish. This can help form a protective coating around the skin and gills. Formalin baths are another means of treatment which can be used in ornamental fish and food fish. However, for food fish, only approved formulations can be used. Typically, you would use a 37% aqueous solution of formaldehyde with 10% to 15% methanol. Do not use 10% neutral buffered formalin, which is meant for preserving tissues for histological section. Formalin baths are used to treat external infections, involving protozoa and metazoa like monogeneans. Although external infections involving oomycetes and bacteria can also be treated with formalin baths, there are better alternatives available. Formalin baths are also used to treat egg infections involving oomycetes and fungi. It's important to keep in mind that formalin reacts with dissolved oxygen. Every 5 mg per liter of formalin removes 1 mg per liter of dissolved oxygen. For this reason, when you're using a formalin bath, you should also provide supplemental oxygen. It's also worth noting that formalin is a human health concern. Formalin is a carcinogen, so make sure you're wearing personal protective equipment and using it in a well-ventilated area. Liming is a very traditional means of parasite control, which can be used in food fish and ornamental fish, specifically in pond aquaculture. Lime is used to remove ectoparasites. Typically, it's just added to the environment. It's also used to maintain alkaline or near alkaline pH. Copper sulfate is another means of parasite control. This can be used to treat ornamental fish for ectoparasitic infections, specifically ornamental fish in an aquarium setting. Typically, copper sulfate is added directly into the aquarium environment. However, it's worth noting that it's extremely toxic to plants and invertebrates. It also disrupts nitrifying bacteria, thereby increasing the levels of ammonia and nitrite. For this reason, it's best to isolate the affected fish before treatment. Also, keep in mind that copper sulfate has a narrow margin of safety, so know your concentrations and keep a close eye on the fish undergoing treatment. Other parasiticides used in ornamental fish include metronidazole, praziquantel, and fenbendazole. Metronidazole is used to treat protozoal infections, both internal and external. Examples of internal infections include spironucleus. Examples of external infections include Ichthyophthirius multifilus. Metronidazole is typically administered into the environment. Praziquantel is typically used to treat platyhelminthic infections, both internal and external. Internal infections may include trematodes and cestodes. External infections include monogeneans. Praziquantel is typically administered as a bath treatment. Fenbendazole is used to treat nematode infections. These infections are typically internal. Examples include Camellanus, which is an intestinal parasite. Fenbendazole is administered via a bath treatment. In Canada, there are only a handful of parasiticides that are approved for use in food fish. Azamethopos is an organophosphate that's used to treat ectoparasitic infections, specifically arthropods. This drug will target adult and pre-adult stages of salmon lice. Azamethopos is a neurotoxin, specifically an acetylcholinesterase inhibitor. It's typically administered as a bath treatment. One of the advantages of this drug is that it has a rapid pre-slaughter withdrawal time of 24 hours. It also has low bioaccumulation. As of 2002, the use of this drug has been discontinued in Canada because of increased drug resistance. Teflubenzuron is also used to treat ectoparasitic arthropod infections. This drug is a chitinase inhibitor, so it specifically targets the molting stages of salmon lice. It's less effective against adult stages. Teflubenzuron is administered as an in-feed treatment. One of the issues is that it has low bioavailability, sitting at around 10%, which means 90% of the drug is excreted into the environment. The image on the top right is an illustration of the chitin subunit. The image on the bottom right illustrates the development of the salmon louse. Emamectin benzoate is an avermectin more commonly known as slice. This is used to treat ectoparasitic infections, specifically arthropods. This drug is effective against all of the life stages of the salmon louse. It's a neurotoxin that specifically targets glutamate-gated chloride channels, causing paralysis of the neuromuscular system. Amamectin benzoate is administered as an in-feed treatment. When used appropriately, it has no pre-slaughter withdrawal time. Hydrogen peroxide is also used to treat ectoparasitic infections, specifically arthropods. However, it can also be used to treat fungal and oomycete infections. It's a common means of treating eggs and hatchery fish. Hydrogen peroxide is an oxidizing agent. It functions via the peroxidation of cell membranes and in causing a loss in enzyme function, specifically those involved in DNA replication. In the salmon louse, bubbles will form in the hemolymph, causing mechanical paralysis. Traditional methods of treatment involve surrounding a pen with tarp and administering the medication in situ. However, some of the lice recover and reattach themselves to the host. Hydrogen peroxide has a half-life of approximately 7 days, so following treatment, it will break down in the environment. A number of concerns have been raised about the effects of hydrogen peroxide on the environment and the potential for the recovery of a portion of the treated lice. In response to these concerns, new methods of treatment have been developed. There are wellboats from Norway that are specialized for delousing farmed Atlantic salmon on board. The wellboats draw up the fish from the pen, treat the fish with hydrogen peroxide, and then release the fish back into the pen. The system can treat 50,000 fish at a time. The treatment takes 20 minutes. The entire process is approximately 2 hours. This method will remove 90 to 95% of the lice, and unlike traditional methods of treatment, the lice are captured and incinerated. Treating the fish with hydrogen peroxide is stressful. Specifically, it causes oxidative stress. So one of the advantages of the wellboat system is that it minimizes handling stress. The wellboat is also equipped with a UV filter for treating wastewater, thereby killing viruses, bacteria, and other pathogens. It also separates bycatch, which are returned to the sea. The wellboats can also be used to transport fish from the hatchery to the open net pens, with a capacity for approximately 250,000 fish. Non-chemical methods of delousing include the thermolyser, the hydrolyser, and cleaner fish. With the thermolyser, the salmon are drawn up into a mobile treatment facility. The fish are then treated with lukewarm water. The lice detach, and then the lice are collected and destroyed. The hydrolyser is a similar system. However, instead of using lukewarm water, the facility uses low-pressure water jets in treatment chambers. The lice detach, and then the lice are collected and destroyed. In regards to cleaner fish, there are a number of species that feed on salmon lice, including lumpsuckers and ballan wrasse. These fish have voracious appetites and can consume up to 175 lice per day. At present, there is ongoing research into better understanding their biology so that they can be farmed effectively. Research topics include habitat, reproduction, and disease. Antibiotics are used to kill or inhibit pathogenic bacteria. Typically, they are administered via medicated feed. However, it can be difficult to treat an inhabitant fish. In regards to global use of antibiotics, different nations use them for different reasons. Examples include the use of antibiotics to treat active infections, to treat fish in quarantine, to counteract the stress of handling, as a substitute for poor hygiene, and in the absence of biosecurity. Many organizations recognize the need to curb irresponsible use and to find alternative means of ensuring fish health, like vaccination and improved husbandry practices. One of the issues associated with antibiotic treatments is that in-feed antibiotics and their metabolites are often excreted in the feces and urine. Depending on the drug used, the percentage can vary between 30 and 90%. This can result in a buildup of residues in the surrounding environment. These residues can alter the microbiota in the sediments. They can also alter phytoplankton and zooplankton diversity. This can have an impact on the food chain. Antibiotic residues in wild fish are also a human health concern. Issues include adverse drug reactions, like hypersensitivity reactions, chronic toxicity, and altered flora. There's also the issue of antibiotic resistance in clinically important bacteria. In regard to global usage, Vietnam and China use the highest number of antibiotics, with 39 and 33 respectively. Only a fraction of these are approved for use in aquaculture. Strict residue requirements on imports help curb the excessive use of antibiotics. There's been a recent shift away from nitrofurans in Southeast Asia, given their carcinogenic properties. More specifically, they cause tumors in the urinary system. The illegal use of banned antibiotics is an ongoing issue. However, fish from problem areas like Vietnam and China are frequently tested. In Canada, there are only a handful of antibiotics that are approved for use in aquaculture. Erythromycin, which is a macrolide, is available by prescription on emergency drug release. Treatments are provided via in-feed administration, and it's used to treat marine finfish. Florfenicol is an amphenicol. It's available by prescription as an in-feed treatment. It's used to treat marine and freshwater finfish. Diseases treated with florfenicol include Aeromonas silmonicida. Oxytetracycline is a tetracycline that's listed within the medicated ingredients brochure. The treatments are provided via in-feed administration, and they're used to treat marine and freshwater finfish. Diseases treated with oxytetracycline include Aeromonas silmonicida, Renibacterium salmoninarum, Yersinia ruckeri, and Flavobacterium. Sulfonamides are available by prescription. Treatments are via in-feed administration, and they're used to treat marine finfish. Diseases treated with sulfonamides include Aeromonas silmonicida, Vibrio anguillarum, and Flavobacterium. Other treatments you may come across include surfactants, which are used to break down excess mucus on the gills to avoid respiratory distress. Malachite green can be used to treat ornamental fish for fungal and parasitic infections. However, this is a carcinogen. Although it's sold in pet stores, it should not be distributed by veterinarians. There are also a variety of water conditioners that are available in pet stores. Most of these are used to treat tap water by removing chlorine, chloramines, and heavy metals. They also help to form a protective coat around the skin, which promotes wound healing. Some of the water conditioners are biological filter additives, so they're essentially a concoction of beneficial bacteria which help break down organic debris and other pollutants. They also promote the growth of nitrifying bacteria. The most commonly used anesthetic is known as MS-222, or tricaine mesylate. MS-222 is a white powder that's added to the water. In order to minimize drug use, it's best to transfer the fish into a smaller volume of water. The anesthetic is fairly acidic, and it can irritate the fish. For this reason, it's important to combine it with a sodium bicarbonate buffer. For every gram of MS-222, add 2 grams of sodium bicarbonate. Anesthetized fish should continue to ventilate their gills. If the fish are no longer ventilating their gills, the dose is probably too high. Euthanasia is achieved with an overdose of MS-222. In order to recover the fish from anesthesia, place the fish into untreated water. You should monitor the fish until it can maintain sternal orientation. Ram ventilation can be used to hasten this process. Assuming you're not dealing with a shark or some other toothy fish, ram ventilation is achieved by putting a finger in the corner of the fish's mouth to force the mouth open. It also allows you to check for increased jaw tone. Once the fish's mouth is open, torpedo the fish around in a figure-8 pattern. Not only can this be used to speed up recovery, it can also be used to reverse excessively sedated fish, which are unable to ventilate on their own.